Good afternoon, guys. I am Dr. Rajendran, Associate Professor of Botany, working at Thiru Government Arts College, Virthachalam. Now I would like to speak about the subject genetics. As the University of Annamalai is concerned, it is one of the subjects under the paper code 21 BOTC 203. It of course it is comprising of cell biology, genetics and plant breeding. And I would like to talk about the subject genetics. As the subject of genetic is concerned, it is one of the oldest subjects of science, and of course it is an applied one in the sense, though it is comprising of a conventional, it is the base for all the applied fields of genetics and genetic oriented fields, the so called genetic engineering and molecular biology and so on. Now I am going to talk about genetics directly. The word genetics derived from the Greek word genetikos. For the first time in the world, the term genetics was introduced by an English genetist, William Batson. Then the word genesis is involved, which means origin, right. So, as the nature of genetics is concerned, it deals with both heredity and variation. I am telling again, as the nature of subject genetics is concerned, it deals both with heredity and variation, not only heredity or not only variation, but heredity and variation. So, as the word indicates, heredity is nothing but a transmission of hereditary character from one generation to another, or parental generation to offspring. Variation through the mechanism or the mode of genetics, variation brings about in the generation of offspring from the deviation of parental generation. So, as the consequence development of genetics all over the globe, we know the father of genetics is Gregor Johannes Mendel, the so called G.J. Mendel. Even before his lifetime, there was a genetics, but no one was in a position to reveal the facts and ideas with stern evidence. But Mendel could reveal the genetics facts, ideas, results with a stern manner in way of evidence, scientific proof. For that, his study was the base. He could in a position to reveal all the things in experimental manner and in the results manner. But the thing is, during his lifetime, his ideas, his facts and results were not accepted by anyone. So that at the end of his lifetime, he was used to say, my day will come. That day came during 1990 year all over the world as the future of non-Mendelian genetics, but now I would like to concentrate on Mendelian genetics. As Mendelian genetics is concerned, it comprising of two things mainly. One is a monohybrid class and another one is a dihybrid class. So, I said you earlier, as the nature of genetics subject is concerned, it deals both with the heredity and variation. I revealed what is heredity and what is variation from the genetics point of view. Whatever may be the thing that we dealt or we are dealing or we would like to deal, the one of the basic component of genetics is genetic material, that is nothing but a DNA for the most of the case, because there is another genetic material that is called a RNA, as in the case of a virus. But for the most of the organism in the world, DNA is the genetic material which is responsible for all the events of genetics. It may be concerned under Mendelian genetics or non-Mendelian genetics, or in the name of a modern or molecular genetics. It is all concerned with the so called genetic material DNA. Before going to deal what is DNA and other things, first we should know the base and fundamental for all the things. So, in this way, for all the things, for most of the organism in the world, cell is the base and fundamental unit, in which there are two main components, as in the case of eukaryote, that is cytoplasm and nucleus. In the case of prokaryote, the nucleus is absent. When we are talking about the eukaryotic cell, then it has two portions, as I said you now. One is a cytoplasmic area and another one is a nucleus area. What I am telling is the nucleus area is purely or directly concerned with the genetical aspects, in which we are having the genetic structure called a chromosome. The chromosome is nothing but a structure consisting of the orientation of a genetic units, that is nothing but a DNA. In the case of DNA is the genetic material. So, simply saying, DNA which is present in chromosome, chromosome is present in nucleus, the nucleus is present in cell. So, this is the base and fundamental structure. Now, DNA is nothing but the sequence of nucleotides. Sequence of nucleotides, they are the components of pyrin and pyrimidine. The orientation is adenine, thymine, cytosine and guanine. So, all these things are constituted DNA, which is responsible for the transmission of characters or brings about the variation from one generation to another. And another term which is used to say regard with genetics is gene. So, gene is nothing but a short the short sequence of nucleotides or a short segment of DNA. So, what is the stressed one is, gene is DNA, but not all the DNAs are genes. that should be kept in mind. Gene is DNA, but not all the DNAs are genes. When we are saying genes, of course, it is nothing but a DNA for most of the cases. As the definition is concerned, DNA is a short nucleotides of a nucleotide sequence or a short segment of a nucleotide sequence. We should have the codes for the synthesis of a specific protein, and also it should carry the genetic information from one generation to another, right. Among the aspects of two things, that is, one is heredity and another one is variation. Among the two events, one is meiosis and mitosis. Meiosis is mainly responsible for the variation, brings about the genetical factor from one generation to another, because during the meiosis the chromosomes are getting apart and making union with the other genetic material, so that we are having new combination with which gametes are produced. By the union of gametes, they are all distinct in its future, then we are having the new offsprings in terms of genetic variation. So, in this way we are getting, or meiosis is the base and fundamental scientific events which brings about the variation in the offsprings through the mechanism of genetics. So, again, among the two things, one is inheritance and another one is variation. Simply saying inheritance is nothing but a transmission of a genetic characters from one generation to another, or in simple term, from parental combination to offspring. When we are talking about Mendelian genetics in strict sense, he did two aspects of experiment. One is called monohybrid. Monohybrid is nothing but the taking of a single character, that is the base of monohybrid class. As per the Mendelian genetics, the single character is a H. Before going to discuss what is monohybrid, what was his experiment, what was his result, what was the law proposed, and what is a test class and what is a back class, what is a phenotypic ratio and genotypic ratio, we should know some of the base and fundamental aspects in terms of structure or in terms of a definition. Among all these things, we discussed two things. One is chromosomal structure and another one is what is DNA, like that. But there are something as far as chromosome is concerned, as in nature case, they are in paired structure. That means in each and every organism, as well as in each and every somatic cells, they are having paired structure. That means they are identical in its nature, in its morphology, in its function, in its physical characters, in its chemical characters. So, right from physical, right from chemical, right from orientation, right from base structure, fundamental aspects, functions, those who are having identical, then that type of chromosomes are paired together, called homologous chromosomes. Among the homologous chromosome, one is the paternal origin and another one is maternal origin. So, homologous chromosome that is more important, that is most important one. Homologous chromosome, a pairing one in which two identical chromosomes are paired together. The one more thing regarding the homologous chromosome is alleles. Alleles are nothing but a pair of genes located on the same loci of the homologous chromosome and controlling the single character Suppose regarding with the monohybrid cross, Mendel took a single character for his monohybrid cross, that is nothing but height. The height of the character has its two forms or has its two traits, that is nothing but tall and dwarf. The gene responsible for tall as well as the gene responsible for dwarf is located on the same loci of homologous chromosome and controlling their alternative characters. So, in this way, in the monohybrid class of Mendelian genetics, Mendel took a single character, as I said you now, the so called height character, and its corresponding opponent traits, tall is on one side and dwarf is on another side. So, in this way he had a tall on one side and dwarf on another side as a parental combination, but they are in a homozygous nature. This is another terminology which is used. Homozygous is nothing but all are pure, all are pure in the sense physico, chemical and genetical. So, in this way on either side he had a homozygous nature of characters. So, in this way homozygous tall on one side and homozygous dwarf on another side. Usually in genetical patterns, the tall or the dominant is not a tall one, but dominant character is denoted with the caps representation. I mean regarding with the alphabet usage, the caps alphabet is used to represent the dominant character, whereas the recessive characters are represented by the small letter of English alphabet. So, in this way the monohybrid case, tall is denoted by caps T and the dwarf is denoted by small t. As I said to you earlier, I mean in the starting point of a discussion, for the segregation of gametes, the meiotic cell division is responsible. So, in this way during this meiotic process, both the genes, the caps T and the small t are getting apart, forming two gametes on each side. But as a rule of genetics, when the genes are identical, at least in two in numbers, when the genes are identical, at least two in number, I mean identical genes, then we have to take only one as representation. So, in this way for two caps T we have to take single caps T. On the other side for two caps I mean small t, we have to take one small t as a representation. So, in this way on one side caps T gene and another side small t gene. During the fertilization, these two genes are united together and forming a zygote. The zygote leads to the development of fully developed offspring. So, in this way we are having the first filial generation, the so called F1 generation. As per the Mendelian genetics of monohybrid cross, the F1 of monohybrid cross is heterozygous tall, because they are the union of a dominant as well as recessive characters. So, in this way they are called heterozygous tall. Here one thing would like to state, in those days there was no much advancement of scientific method to analyze or to prove or to standardize the results or to reveal. The basis at the time to reveal the experiment or to reveal the results, what were the basis or means purely of morphological characters, and to some extent only cytological, but most of the cases to reveal the results, morphological characters were the basis. So, in this way during his monographical class, Mendel took homozygous tall on one side and homozygous dwarf on another side. I means one is dominant, pure dominant, and another one is pure recessive, but he had a heterozygous tall, on which or on the basis of the results people used to represent the law, that is nothing but a law of dominance, but it became failure one, or it not at all a standardized one. Law of dominance is not a standard one or became a failure one, because in the next generation, recessive genes are also having their influence in the prediction or in the expression of or in the inheritance of character. So, what was the base to say the law of dominance is, on one side they had homozygous tall and another side they had homozygous recessive, but in the F1 they had a heterozygous tall. Then they came to assume only dominant genes are having their power to express, not recessive genes. So, this was the base idea to propose the law, but in the F2 generation it became failure, because in the F2 generation they are having phenotypically two types of results, one is a tall and another one is a dwarf, but genetically they are having four types of combination. One is homozygous tall, two are heterozygous tall, and one is a homozygous recessive dwarf. The possibility of homozygous dwarf is the segregation or getting apart of recessive gene from the dominant gene. So, in this way from each generation or from each combination, the recessive small t gene is segregating apart, and where they are getting chance to reunion, they are reassembled, so that we had the chance to get the trait of a homozygous recessive dwarf. So, as in the case of F1, the law proposed was law of dominance, it became a failure one or otherwise not a standardized one, but the standard one is the F2 results. Based on F2 results, they are proposed the law, that is nothing but a law of segregation. What is law of segregation is from the combination of caps t, small t on one side, similarly on the other side also caps T, small t. From the both the cases, the small t's are segregated or getting apart, and they got reunion when they are getting chance. Only that we could have the homozygous recessive dwarf. So, due to the segregation of a recessive gene from dominant, not only that, when they are getting a chance of reunion, they got reunion and expressed a trait, the dwarf trait, that too in the manner of a homozygous, not a heterozygous one, but homozygous one. So, in the genetic segregation, all are possible, not only the law of dominance, but all are possible. So, as far as the monohybrid cross is concerned, it is considered. Which one is considered is a law of segregation is considered, and the law of segregation is accepted. But in the case of a law of dominance, it became a failure one, again I am telling. Right. So, here to prove two things, we are having some sort of a continuous patterning of crosses, that is nothing but a test cross and back cross. Generally, they are all in usage and they are all in practice, but they are familiarly discussed with the Mendelian genetics with a special reference to monohybrid and dihybrid. The crosses are test cross and back cross. Test cross is to test, right. So, let us talk something about a test cross of a monohybrid cross. What is it is? There are some logic. What is the logic is? It is to test. What we are going to test? Again, in the character production or in the inheritance of a character, whether the role of a dominant gene is present or whether the role of a recessive gene is present or both. So, this is the questions. Again I am telling, in the character production or in the inheritance of character, whether dominant has its role or recessive has its role or both. This is the logic behind in testing. Why is it formed? Why is it developed? So, when we are going back to the cross of monohybrid, Mendel had pure tall on one side and pure dwarf on another side, but he had phenotypically tall in the F1. Genotypically they are different, but no one was known. On seeing or on the assessment of phenotypic, people got assumption, that is dominant genes are having their role in the character or in the trait production, but it is not so, as we discussed earlier. But anyhow, to test whether this one or that one or both. So, in this way they had to test the role of genes. So, in this way they have formed a logic for test class. In this way, for doing test class, the F1 should have crossed with the recessive parent only, right. So, for the test grass, the F1 should be crossed with the recessive parent only. So, in this way the F1 is caps T small t. Then the recessive is small t. The cross is made between them, that means caps T small t on one side and small t on another side. Here there are two gametes are produced through the meiosis, one is caps T and another one is small t. On the other side, two small gametes are produced, that means small t. Crosses are made among them, After getting the cross, we had two types of results, one is tall and another one is dwarf. So, the result percentage is 50-50. It is represented as 1:1. It is represented as 1:1, right. So, the aim of test grass is to test the role of genes, whether the dominant has its role or whether the dwarf is has its roles or both. Here, to be frank, both the genes are there having their role in the expression of a trait. So, here it is proved, 1:1. On the other hand, backcross, as a definition is concerned, the backcross is crossing of F1 with that of any one of the parental combination. Here we should mind some sharp deviation. As the definition of backcross is concerned, crossing of F1 with either one of the parents, it may be of a tall one or may be of a dwarf one, but in practice, I mean during the recent days, the aim of backcross has been changed. The aim is to get the superior one at every time, at every time. This is the aim or the logic of doing backcross, but it is possible when the F1 is crossed with the superior parent only, not with the recessive parental. But the literary form of a definition is, what is backcross? Backcross is nothing but a crossing of F1 with any one of the or either one of the parental combination. It comprising of both recessive as well as dominant. But the interest of backcross is getting superior trait in every generation. Suppose if you have to do it, if you have to acquire the superior characters at all the times, then we should cross or we should make a cross between the F1 with that of the dominant parent, only, not with the recessive parent. But the definition is insisting like that, any one of the parent, but it should not be. To get the desired characters in generation to generation, the F1 should be crossed with the dominant parental. Only then we could have the superior characters at every time. So, in practice, what is backcross? In practice, not literally, but in practice, the backcross is having crossing of F1 with the dominant parent. But in the case of test grass, no alteration at all. Test grass is to test which gene has its role, whether dominant or what do you call recessive or both. So, this is the aim of backcross and test grass. So, as far as the monohybrid cross of Mendelian genetics is concerned, only single character is taken for cross, that is mono, single character, but it has its contrasting forms or contrasting trait. So, the character 8 has its contrasting forms, that is nothing but a tall and dwarf. So, this is a thing said or discussed earlier. So, as a conclude, the monohybrid class is coming under the Mendelian genetics or the classical genetics, in which single character was taken for crossing. The single character is a height, but it has two contrasting forms, one is tall and height as a parental combination. As Mendel is concerned, he had a pure form on both the sides. So, in this way he had pure tall on one side and pure dwarf on another side, having mixed tall, the so called heterozygous. Then he make a self cast for getting F2 generation. Here I would like to state one thing. In the regular practice or rules of genetics, to get the F2 from F1, one should go for self cross of F1 to get the regular F2, not with any deviation of ideas. In general, to get the F2 from F1, one has to go for self cross. So, in this way the F1 of a monohybrid had a self cross. At the end of F2, we are getting two phenotypic results, that is nothing but a morphological oriented one, in which as a phenotypic result is concerned, we are having two types of traits, one is tall and dwarf. But when we are so in the ratio of 3:1, that means 3 tall and 1 dwarf. This is about phenotypic ratio. But the progenies are 4. Among the 4, one is dwarf and other 3 are tall. When you are going for genetical analysis, one tall is having pure form of genetics and 2 talls are having the mixed type of genetics. So, the pure is called homozygous tall and the mixed type of genes which are responsible for tall character is called heterozygous tall, whereas in the case of dwarf it is pure form, right. So, as the phenotypic ratio of monohybrid class is concerned, it is a 3:1 phenotypic ratio of a monohybrid class, whereas the genetical cross of a monohybrid class is 1:2:1, that is 1 homozygous tall and 2 heterozygous tall and 1 is homozygous recessive. And the other part of a conclusion is having of a test cross and back cross. Test cross is to test whether dominant gene has its role in the character expression or the recessive gene is having its role in the expression of a character. But the result is both the genes are having the chance of revealing or the inheritance of characters. Inheritance of characters, that is proven by test cross. And another type of cross is backcross. Backcross is nothing but the crossing of F1 generation with either one of the parental combination. This is a literary definition, but in practice, to get the superior variety or superior trait from generation to generation, one has to cross the F1 with that of the dominant parent only. Only then we can have the superior or the dominant trait or character in successive generation. So, in this way, the test cross ratio is 1:1, 50% of dominant and 50% of recessive, or 50% of character is one kind and another 50% of character is another kind. So, this is what you call about monohybrid class of Mendelian genetics, right? So, at the end of F1 generation, what was the law proposed is law of dominance. That is at the level of F1 of a monohybrid class, because against the characters taken, that is a homo tall on one side and homo dwarf on another side. The progeny has the mixing of genes. But it was believed that always dominant gene has its role in upper hands. That's why it was proposed. But for in the case of backcross, as we said, to get the superior character way, as the ratio of a phenotypic ratio of a monohybrid class is 3:1, and the test cross ratio is 1:1. Phenotypic is 3:1. Genotype of F2 is 1:2:1. One is homozygous tall and two are heterozygous tall, and one is homozygous dwarf. So, this is about the monohybrid class. Since Mendel had mathematics and natural history as his subjects of expertise, he could in a position to reveal his genetical experiments results in authentic manner or in proven manner. In his continuation, he went for the another genetical cross or research, that's nothing but a dihybrid cross. So, just like of a monohybrid cross, in which he had a single character, as we discussed, the single character was a 8, but for parental combination he had two different forms of 8 that we dealt. Likewise, here in his dihybrid class, Mendel took two characters. For his experiment. I am repeating it. When we are talking about the dihybrid class of a Mendelian genetics, Mendel took two characters for his research. What were two characters? The two characters were shape of the seed and structure or orientation of the seed. Color. One is orientation or shape or structure. These are all on one side of the seeds, and another criteria is color of the seed. Color of the seed. So the base for dihybrid cross of a Mendelian genetics is two characters. The two characters are, one is a shape of the seed and another one is a color of the seed. One is shape of the seed and another one is a color of the seed. Color of the seed. Of course, each and every character has its contrasting forms or trait. So in this way, the shape of the seed has its two forms. One is round and smooth and another one is wrinkled. One is round and smooth and another one is wrinkled. On the other hand, the second character is color of the seed. Color of the seed. So one is a shape of the seed and another one is color of the seed. Color of the seed. So in this way, in his dihybrid cross, Mendel took two characters. One is shape of the seed and another one is color of the seed. Of course, each character has its two contrasting forms. In this way, round and smooth type of seed on one side and round, smooth and green color I'm sorry, yellow round. Yellow, the color is yellow. A round is the shape. So on one side, Mendel had a round and yellow. Round is the shape of the seed and yellow is the color of the seed. On other hand, he had another character that is also a shape of the seed and color of the seed, but the things are deviant one or the contrasting one. Here around the seed, there wrinkled type of seed. Here round the seed, there wrinkled type of seed. Whereas the color is green. So these are the two contrasting forms of character. So as I said to you, now two characters, one is a shape of the seed and another one is a color of the seed. So in this way, one parent has a round shaped seed and yellow colored seed. On the other side, another parent has a wrinkled or incomplete type of, shape of the seed and green color. Here, among these two characters and their contrasting forms, round and yellow seed is considered as dominant one. Considered as a dominant one, whereas the seed with the green color and wrinkled shape is considered as a recessive one. As a recessive one. Moreover, they are represented. They are represented by the English alphabets. By the English alphabets. Caps or stand for round, and caps yellow stand for yellow color. This is the gene combination on one side. Caps R and caps Y. On the other side, small r and small y. This is the representation of recessive characters. So, caps Y, caps R. This combination is the representation of a dominant character. That is nothing but a round and yellow seed. Whereas on the other hand, small y, small r, or small y. This combination is the representation of recessive characters. The characters are wrinkled seed and green color. Right. The cross is made between them. And we are getting F1. So, but the F1 is in heterozygous form. The phenotypic expression is yellow round only. Of course, phenotypically they are dominant one, but genotypically they are in mixing form or in mixed form. That is nothing but caps Y, small r and caps R. This is the state of genotypes of dihybrid. So, as a dihybrid class of Mendelian genetics is concerned, he took two characters for his analysis or for breeding. On one side, he had a pure breed of two characters, that is a round shaped seed and yellow colored seed, represented by caps R and caps Y. This is considered as dominant one. On the other side, he had a wrinkled seeds and green colored seeds. Represented with a smaller and small y, which is considered as a recessive one. Of course, crosses are made between these two combinations of genes. In the F1, Mendel got, of course, phenotypically a dominant one, that is nothing but a yellow color and round shaped seed. At F1. Of course, phenotypically they are dominant. Phenotypically, that is externally. But genetical analysis reveals they are heterozygous. That means For round shaped, there are two types of genes. One is dominant genes and another one is recessive genes. And for the color, there are dominant gene of a caps y and the recessive gene of a small y. They are all representing for F1 of a diabetic class. By all these means, the genetical analysis revealed heterozygous, though they are dominant for looking. Right. So, as usual, what do you call he made a self-class among the F1 for getting F2 generation. So, this is the general rule of genetics to get the F2 to get the F2 from F1. That is nothing but a self-class. In this way, Mendel made self-class of a F1 of a dihybrid class. Right. Here we would like to say two methods for carrying out the F2. One is a forking method and another one is a Punnett square method. Forking method, when the genes are limited and the flow is simple and the results are simple, then one can go for forking method, as in the case of a monohybrid class. But when the genes are more in number and they are complicated to predict the results and the segregation, it's better go for Punnett square or checkerboard. So, in this way, the F2 of a diagrams is predicted with the help of a checkerboard method or the Punnett square method. So, in this way, we are getting 16 offsprings in F2, with different combinations, of phenotype and genotype. Simply saying, out of 16, we are getting 9 superior results as that of one side of parent, and another one is a recessive results as one side of parental combination. That is 9 of yellow round and 1 of green wrinkled. The remains means 16. Out of 16, they are comprising of 10, and the remaining 6 are deviant. Among 6, 3 are the mingled form, that is nothing but a wrinkled yellow, and another 3 are round green. So, these two things are contrasting from their parental combination. So, again, for getting clarification, in the F2 of a dihybrid class, one can have 16 offsprings out of parental combination through the way of self of a F1. Among the 16, 9 are of with one parent and 1 is with another parent, and the remains are different from parental combination. In this way, 9 phenotypic is round yellow and one is wrinkled green, and the remaining 6 are mixed form. So, in this way, round green, wrinkled yellow. How is it possible? Where we can means not ourselves, but genetics can produce another law at the level of F2 of a dihybrid cross. That is nothing but independent assortment. Independent assortment. It holds its accordance still now. Keep in mind, guys, as the law proposed for dihybrid class of Mendelian genetics, that is nothing but independent assortment holds its accordance even now. That means highly standardized one, independent assortment. How is independently assorted? So, we are having 4 genes to express 2 characters. People thought the genes are always united together and segregated together and inherited together. For the sake of understand, I repeat the sentence. People thought, even scientists and geneticists thought, always alleles are united together, traveled together, segregated together and inherited together. This was the strong belief. But it's not so. Whenever they are chance, genes are tend to apart or segregate apart. It's all depending upon the chances. When chances are there, genes are getting apart through the segregation, through the meiosis, through the meiotic segregation. So, in this way, the gene of wrinkled, getting apart from green color. Similarly, on the other hand, the genes of a color rounded is getting apart from the the genes of yellow color getting apart from the genes of round shape. So, at one stage or to a certain stage, when the chances are not having the combination genes are united together and inherited together, but when they are having a proper chance, they are getting apart, or in other words, the alleles are getting apart. So, in this way, one could have a new combination which are not present in the parental combination, because as far as dihybrid class is concerned, one parental combination is yellow round and another parental combination is green wrinkled, but at the end of F2, a round is separated from yellow and the green is separated from wrinkled, and they are making a new combination. Through the way of making new combination, the genes, the yellow colored genes are pairing with wrinkled shaped seeds. Similarly, green colored is combined with round shaped. That's why 2 new combinations are produced. New 2 combinations are produced. That is nothing but yellow wrinkled and green round, which are not present in the parental combination. They are newly combined one. It's all because of a independent assortment of genes. What does it mean is, whenever the genes are getting chance to apart from its combination or apart from its alleles, they are getting, and similarly whenever they are getting chance to combine with a new gene, they are combined. So in this way only, here, I mean in the case of F2 of dihydric acid, we are getting two more new combinations of traits, two more new combinations of traits, that is nothing but green round and yellow wrinkled. It is because of the independent assortment of genes from its pair or alleles or combinations. That's why. So in this way, the phenotypic ratio is 9:3:3:1. 9s are yellow round, 1 is a green wrinkled, remaining 6, 3s are yellow wrinkled and 3s are green round. Again, as in the case of monohybrid cress, researcher mad back and test crosses, the same purpose, to test whether the role of a dominant or whether the role of a recessive in the character expression. Likewise, backcross to get the superior traits or to get the superior characters from generation to generation, then one should go for backcross. As in the case of a test cross of a dihybrid, we are getting 4 different combinations of genes with a single representation. That's why 1:1:1:1. That means 4 ones. 4 ones are produced as the result of a test cross of a dihybrid cross. So as a conclude of a Mendelian genetics, conventional one or the traditional one, under Mendelian genetics, Mendel made 2 types of crosses, one is a monohybrid cross and a dihybrid cross. In both the cases, he had a pure type of combinations of genes for crossing. In the monohybrid cross, he had a single cross, a single character, that is H. Whereas in the case of a dihybrid cross, he had 2 characters for his crossing, that is nothing but one is the shape of the seed and another one is the color of the seed. In both the cases, in at the F1 level, he had the dominant characters like that of the parental one phenotypically, but genetically they are different one. And for getting F2, he went for a self cross and he had results. Of course, in each cases he had different results also in the way of phenotypic and in the way of genotypic. It's all because of segregation of genes. When they are getting chance. In this way, law of segregation law was proposed for F1 monohybrid cross, and a law of independent assortment was proposed for dihybrid cross. among the laws proposed, the law of independent assortment has or holds his accordance even now. Even now. That means highly standardized one. Highly standardized one. So, in this way, Mendel laid a strong platform for the real scientific study of genetics and the understanding of some basic and fundamental principles regarding the function of genes, based upon which only the other genetics have been built up in the name of non-Mendelian genetics by others after his or after Mendel's lifetime. And in the current century, the genetics has its a new form of expression or discussion with the name of a modern genetics or with the name of a molecular genetics. It is purely based upon molecular level of reveals. In strict sense, they are at the nucleotide levels. Let us discuss in the continuous program. Thank you, one and all. Thank you very much.